His studies of the apical ectodermal ridge, or the AER, helped to identify the role of growth factors in the growth and patterning of developing limbs, and his identification and study of the zone of polarizing activity, or the famed ZPA, demonstrated how this regionally limited tissue spreads essential information across the limb to dictate different skeletal patterns. His early recognition of the role of cell death in developmental morphogenesis and how developmental fields are set up to make intricate feather patterns are also part of his long legacy to scientific understanding of limb development. John Saunders was a member of the National Academy of Sciences, a past president of the Society for Developmental Biology. He was a summer investigator at the Marine Biological Laboratory from 1958 to 1972, as well as a lecturer in the embryology course during those years, and also from 1995 to 2003, when I had the pleasure to have met him, and he was just an amazing scientist and a really influential developmental biologist. Cliff Taban is really the ideal person to give the John Saunders lecture, and I'm going to turn the mic over to co-course director David Sherwood to introduce Cliff.
Thank you, Carol. I should just add, I also had the pleasure of meeting John in 2002, I believe it was, and he was just a real gentle giant in the field and a warm and giving person in the lab. So Cliff Taban is the ideal person to give the Saunders lecture. Cliff did his undergrad work at the University of Chicago, where he received a BS in physics. He earned his PhD at MIT working on RAS oncogenes in Bob Weinberg's lab. Still at Harvard, he was a postdoc with Doug Melton. He was appointed to the faculty in the Department of Genetics at Harvard Medical School in 1989 and promoted to full professor in 1997, and became the chair of the department in January of 2007. He has received many awards, including the Conklin Award from the Society of Developmental Biology, the March of Dimes Prize in Developmental Biology. He is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. Cliff has made numerous seminal discoveries on embryonic development and evolution, including work on the secreted morphogen sonic hedgehog in the limb and left-right asymmetry. And his amazing work extends to such questions as the causes and variations in beak shape in Darwin's finches. So it's an absolute pleasure to introduce Cliff. And from there, Cliff, please take it away.
Okay, thank you very much. So do you have, are my slides up now? I can't tell.
No. Can you reshare your slides, please?
Yep. How about now?
Perfect.
Thank you.
Okay, great. So I'm very honored to give the John Saunders Endowed Lecture in Embryology. As was said in the introduction, he's a true giant, and a huge amount of what we do in modern developmental biology, directly or indirectly, is building on his work. So what I'd like to do in my first talk, for this next 45 minutes or so, is tell you something about John Saunders, sort of expand on the introduction that Carol gave, and to explain who he was as a person, how his career developed, what his discoveries were and how he got there, and how those discoveries have led to a modern understanding and what sort of the modern interpretations of experiments might be. Then after the break, I'll give us a second lecture, which will be more research oriented based on some of the work we're doing in my own laboratory now. So to start, John Saunders was, as I said, a pioneering scientist who shaped the field of limb development. But as Carol also said, he was a really devoted educator. He was really committed to training of generations of embryologists, both in his own laboratory and through the Woods Hole course here at MBL. As was said, he was, it's sort of fitting that the embryology course of the MBL continues to benefit from John's legacy through this lecture series. He cared deeply about the course, He participated in 2 different periods in the course. And I also want to thank, before I forget, I want to thank Lee Neiswander, who gave the inaugural Saunders lecture a couple of years ago, who generously shared her PowerPoint presentation with me, and from which a lot of the slides of John, including this slide here, are taken from her. So to understand John, you really need to understand where he came from. He was born into a very poor family in Muskogee, Oklahoma in 1919. His father was actually a barber, and when he was a very young boy of about 4 years old, the barber shop burnt down, and that was their only source of income. And from that point on, he was really a destitute family. From age 8, he was working to support the family. All the money he earned going towards the income of the family continued right through high school. And that was really important in sort of establishing a really strong work ethic that he had. The quote at the top is from John, that he attributed his success to that strong work ethic, followed by his natural intelligence. And he really was a very insightful scientist and a good measure of luck. And you'll see where luck played into a couple of times in his career, but luck only works if you're astute enough to understand what the luck is giving you and you're able to take advantage of it, and he certainly was. So maybe the first piece of luck was he was a really good student in high school, and one of his classmates happened to be a state senator's son. And that state senator was not a very strong student. And the state senator told John if he could tutor his son to pass French, and he was doing some tutoring to make money, that's one of the sources of income. He said, if you can tutor my son well enough to get him through French, I'll fund you to go to college. Now, not only had no one in John's family gone to college, he hadn't been thinking about college at all, but nobody from that high school had gone to college. So the teachers were quite excited about this. And from talking to John, he actually doesn't think he successfully tutored this kid at all. He thinks that the faculty of the high school were so excited about having him go to college that they sort of gave the son a pass to get John through. One way or the other, John was, the son passed French, and John was able to attend the University of Oklahoma, where he got both a master's degree and a bachelor's degree in 1940 and 41. And then he went on to serve in the US Navy during World War II, upon which he went to graduate school at Johns Hopkins University. He actually was accepted at both Harvard and Duke as well, but Hopkins offered him a larger stipend, and it turned out to be a very good choice for him regardless. He worked with a man named Benjamin Willier. Willier was a pioneering experimental embryologist. He used the CAM assay, the chorioallantotic membrane assay of birds, as a site for transplantation studies, studying germ cells, axis formation. Willier published a lot on both adrenal gland and heart and liver formation, et cetera. But one of the reasons Willier was so all over the map in terms of his research is that he didn't tell his students what to work on. When a student joined Willier's lab, he said, come up with a problem. So John did some reading, and he decided to start by trying to understand the formation of feather tracts. He was looking at the back of a bird and seeing these very distinct feather tracts that you can hopefully see here, and in various rows. And he started by doing some sort of rotation experiments, transplanting mesoderm versus ectoderm to try and understand what controlled the feather formation. Now, he actually didn't get very far, as I'll say in a second, while he was a graduate student, but he did come back to this problem when he was later in his career, when he had his own lab, and showed that the mesoderm provides the instructive signal to form the ectodermal appendage, that is where the bud will form. It specifies, the mesoderm specifies whether it'll be a feather or a scale, if the feather pattern is specific to the upper limb or the lower limb, whether it's dorsal or ventral, the polarity of the feather, and so on. And he also could show there was a temporal order to this. The feather tract is established first, followed by the feather identity, that is the specific morphology within the tract, then the direction of outgrowth, and lastly, the dorsal ventral polarity. So he made a lot of progress on this, but that was after he was a graduate student. As a graduate student, he just started these experiments where he was rotating the ectoderm relative to the mesoderm, so he was changing the relative orientation and trying to look at these. And he took his very preliminary data to Willier just to show that these transplants were working. And while he was showing him under the microscope, Willier noted that the dye he was using, which was Nile blue to stain these feather buds, was also staining something at the tip of the nascent limb bud. So at the stage he was looking at the feathers, the limb bud was just starting to form. That's the limb bud. And there was sort of a stripe at the tip of the limb bud. And he said, Willier said, "What's that?" And John said, "I don't know." He said, I've noticed this. There's sort of like a seam where the dorsal and ventral surface come together, sort of like if in your own hand in the embryo, it would be like a little seam running along the tips of your fingers where the palm of the hand meets the back of the hand. And he said there's like a ridge there. It's already been described histologically. It didn't have a name, but Saunders gave it the name of the apical ectodermal ridge. And he thought that was kind of interesting. So Willier said, "Well, while you do the feather experiments, why don't you see while you're trying to fish for something that'll work out to a thesis, why don't you see if you can figure out what this thing is?" Now, modern days, if you want to know what a gene does, you knock it out and say, what happens to the embryo in the absence of that gene? So this was a surgical knockout. He said, all right, well, I want to see what the AER does. Let's see what happens to the limb bud if you remove the AER. Now, if you remove it, the ectoderm heals almost immediately. There's no scar. You end up with a limb bud that looks pretty much the same except without the ridge. All the mesoderm is there. The vast majority of ectoderm is there. And so it was quite stunning that just that slight removal of that little seam gave him a really dramatic result. It had a complete truncation of the distal limb. So if he removes it at a particular stage, he might get the humerus, but he loses the radius and ulna and all the digits. So that immediately was clearly a phenomenon that he could study and make some sense of, or he could try and pursue. So for his thesis work, he did two things. First, he mapped the fate map of the limb bud, which no one had really done carefully before. So using carbon particles, he took a look at how the limb bud develops. And the first thing he noticed was that at an early stage, the relative orientation of the proximal limb is pretty fixed, whereas the distal limb expands a great deal subsequently. So there seemed to be a proximal to distal order in which the expansion of the tissue takes place. And the second thing, this is something that if you ever look at a modern textbook of the limb, it's totally ignored. He's absolutely right about this. At the early stage, if you look at the primordium, what will be the humerus and what will be the radius and ulna, they form at right angles to each other. People always draw them in a linear fashion, humerus, radius, ulna, digits. But in the development, in mammals as well as birds, what happens is the humerus forms at right angles to the radius and ulna. Now, that doesn't really mean that they form at right angles at the time condensation occurs. This is a fate map. What this is saying is that over time, proximal to distal changes in orientation. When the humerus is forming, this is the proximal distal axis. When the radius and ulna is forming, this is the proximal distal axis. It's something that's still not understood really to any extent. It's ignored in the literature, but John was absolutely right about that. The other thing he found which was very interesting, was that if you removed the AER early on, you could lose almost the entire limb bud. Whereas if you removed it later, you progressively lost less and less of the limb bud until at the end, if you removed the AER late enough, you're just losing the tips of the digits. So the two main take-home lessons were the structures of the limb are laid down in a proximal distal sequence, which went against the dogma. That was changed the way people thought. People had thought that the distal elements were established first. And the ectoderm has an essential role in wing development. And that was the most controversial finding, because prior transplant experiments in amphibian embryos had indicated the limb mesoderm would form a limb when grafted under flank ectoderm. But grafted limb ectoderm didn't produce an outgrowth, and which to the previous embryologist said, the ectoderm really doesn't do anything except just hold the mesoderm together. And this was really quite a stunning finding. He presented it at a meeting in 1947 and then published it as his thesis in 1948. But at that meeting, this is not a bad little thesis to have something that sort of totally changes the way people think about development. To sort of emphasize the impact, Paul Weiss, who's a leading embryologist of the day, worked on morphogenesis and neural differentiation. Paul Weiss was also the one who showed with limb regeneration in newts that if you remove the skeleton before amputating, after amputation, the entire skeleton distal to the amputation, that is, the regenerate forms a skeleton without having a skeleton to build on, which says there's some transdifferentiation going on. But anyway, Paul Weiss, who was already a pillar of the developmental biology world, raised his hand after his talk, and when he called on him, he stood up and rather than addressing John, he turned to the audience and said, "Ladies and gentlemen, this is the most significant finding in developmental biology in the last 50 years. So that's not a bad thing to do for your PhD thesis. And he went on after that, subsequently, he and his colleagues, after he had his own lab, to make a lot more progress on the AER. If you graft it to the dorsal surface or the ventral surface, this is a graft to the ventral surface, an entire new limb bud will grow out, saying that the AER can drive out growth. You can graft between forelimb or hind limb or between species, from chick to duck or turtle, and show that the AER still works, so it's not species specific. The two most important findings I think he had subsequently were, number one, this one, that if you graft an AER between young and old limb buds, you get a normal limb. Because one way you could have imagined this was that the signal is changing with time. That the AER makes early makes a humerus signal, then it makes a radius and ulna signal, then it makes a digit signal. But it's not an instructive signal, it's a permissive signal, because if you take an AER from a very old limb bud and put it on a very young limb bud, or vice versa, the limb's perfectly normal. The other thing he found, which I'll come back to in a few minutes, is that if you remove the AER, there's this dark stripe underneath the AER here that he's showing, that if you remove the AER, there's a domain of 150 to 200 microns of cell death. So among the other things that the AER does is it protects against cell death. All right, taking a digression from John for a second, moving into the modern era, people started cloning growth factors, and very quickly, people in a couple labs, Gail Martin's lab and Mario Kopecki's lab, found that members of the FGF family, shown here in an in situ hybridization, so you're looking at the RNA here in purple, of several different FGF family members are expressed very specifically in the AER. And not only are they expressed in the AER, but this was really a stunning result. Both Cheryl Tickle working with, and Lee Neiswander working with Gail Martin, and independently John Fallon, found that a bead soaked in FGF protein can completely substitute for the AER. And that's remarkable for a couple reasons. Number one, at the time this was done, it was already known that the AER makes other factors too. It makes BMPs, it makes WNTs, it's making all of these important signals. And yet the only thing you need to substitute for the loss of the AER and give you a humerus, radius, and all the digits, the only thing you need is FGF. So that was totally unexpected. This was sort of like a shot in the dark experiment that worked extraordinarily well for both of them. And the second thing is the concentration of FGF is not important. So here's a signal, and you put a sort of a random amount in a bead and slap the bead at the tip of the limb bud, and it works. So it's not a morphogenic, carefully titrated concentration. It's just you need FGF around, and if you have FGF around, the limb can grow out. Well, this is a sufficiency argument. The necessity argument, people turn to the mouse. Now, as I said, Gail Martin was one of the people who was involved in this very critical experiment. She also was a pioneer in mouse genetics. And by coincidence, at the same time as the FGF bead experiment was being done by Lee Neiswander in her laboratory, other people in Gail's laboratory were developing the Cre-Lox system for specifically knocking out genes in specific locations. So they actually used the MSX2 promoter, which is an AER-specific promoter that others had defined, as a test case for the Cre-Lox system. And using it as a test case, they found quite dramatically that if you remove several different members of the FGF family, this is a FGF4, FGF8 double knockout that Jin Sun did, you completely lose the limb. So the AER is not only sufficient to drive outgrowth of the limb, FGFs are not only sufficient, they also are completely necessary, and they totally explain what happens when you remove the AER. Now, how the AER functions, what its role is something somewhat controversial. Early on, the first models that were developed to try and explain the patterning effect of this, so how the AER translates to giving you these different limb segments, was what was called the progressome model, which is Louis Wolpert was a proponent of. And the idea there was a timing mechanism. The cells within the mesenchyme, he postulated, are constantly changing from proximal to distal. They start out being shoulder, then they become upper arm, then they become elbow, then they become lower arm, then they become wrist, then they become hand. And they'll do that totally on their own if left to their own devices. But they can only do it if they have a permissive signal from the AER, that is to say FGFs. So early on, everybody sees FGF and everybody's happy and they're all progressing. But after a short period of time, the limb buds expanded and the range of activity of FGFs is limited. It can only diffuse so far. And because of that, the cells that are out of range of FGF are stuck. They can't progress any further, so they become upper arm, whereas the ones down here keep progressing, they become lower arm. And then with more time, some of those get out of range of FGF and they're stuck being lower arm, whereas those that are still within range of FGF can transform into hand or wrist. So that was the progress zone model, which is a timing mechanism. Then Miguel Torres, and I guess to some extent our own laboratory, made arguments for a different model, which involved having two signals, one from the flank and one from the tip. Whereas the limb bud grows out, initially the signals completely overlap. Over time, they pull apart, and you end up with places that see only the proximal signal, the distal signal, or nothing in between. And that was a different way of the way the AER, or the distal signal, could play into this. I think the current model, as best as we can see it, is sort of a hybrid between the two. Miguel Torres is really the one who refined it, but I think there's still a lot of holes in it. I don't think it's by any means nailed down. But the way it's viewed now is there are these two signals, and as long as retinoic acid is being seen by the cells, they're going to be proximal. For example, with this proximal gene MEIS1 and MEIS2, which will form the stylopod. And FGF can't do anything while retinoic acid is there. But as they pull apart, the range of retinoic acid is limited. And the distal cells now see FGF, but not retinoic acid, or initially see maybe a bit of an overlap, but then eventually only see FGF. And once they do, they kick in this progression of Hox gene expression that further subdivides the rest of the limb, not based on the signaling anymore, based on a timing mechanism. So so once this happens, you're on an intrinsic timer, but that timer does not depend on the AER for its progression. The reason the AER is still important at these later stages is because of the cell death. If you remove it at this stage, you're losing the HOXA11 domain, whereas if you remove the AR at this point, the cell death removes the HOXA13 domain. So it's sort of a combination of the two that people currently think of this. So returning to John, he wasn't content to stop with the AER transplants. After graduating with his PhD in 1948, he took a faculty position as an instructor in zoology at the University of Chicago. A year later, he moved to Marquette, essentially because they gave him a loan to give him a down payment of the house. And he wanted a house, so that was sufficient to steal him from Chicago. And he moved to the Marquette University, and he continued to do the AER transplants, as I told you a few minutes ago. But he also did transplants of the entire limb ectoderm, to see whether the ectoderm, the rest of the ectoderm, had any effect at all. And to understand that, you have to understand the orientation of the ectoderm. I'm just going to sort of give you a couple cartoons. If you take the ectoderm off of a left hand, keeping the hands facing up and outward, except at a limb bud stage, obviously not at a hand stage, but when you have limb buds. If you take this ectoderm and put it over this mesoderm, what you've done is you've kept the dorsal ventral axis maintained. Palms are still toward you. The backs of the hands are still away from you. But the anterior posterior axis is flipped. The thumb side is now, the thumb side of the ectoderm is at the non-thumb side, the little finger side of the mesoderm. However, if before doing this transplant, there it goes. If before doing this transplant, you were to turn over the limb bud, so this is the left limb bud, but you flipped it over, and then you transplant it. Now the back of the hand is covering the palm, so now you've flipped the dorsal ventral axis, but the anterior posterior axis is the same. So you have not changed the thumb or the little finger relative to each other. So if you do this first manipulation, changing the AP axis of the ectoderm, you have no effect on the limb whatsoever. But if you flip the dorsal ventral axis, you do have a change. You find that the dorsal ventral axis is totally determined by the ectoderm. So for example, here you can see that the back of the hand has big, large scales, whereas the palm of the hand has tiny little scales in the foot where there's scales rather than feathers. So if you look at just this example, for example, where this flip is done, By the time the flip was done, the more proximal limb has already been determined. It's already been established, and it is developing as dorsal. But the more distal part of the hand is developing as ventral. So you're going from dorsal scales to ventral scales directed by the ectoderm. And the reason that you'd have, I'll come back to in a second, this difference between here and here, the thought is at the time the transplant was done, this part was already specified, but this part had not been established yet. Well, we now know that John was absolutely right about the dorsal ventral axis as he was about the proximal distal axis. We now know some of the genes involved. There's a gene called Wnt7a, which is expressed in the dorsal ectoderm. And Wnt7a is the dorsal signal. It's necessary and sufficient to turn on a gene called LMX1B, a transcription factor that's in the mesoderm. And you can show this if you misexpress Wnt7a throughout the entire ectoderm, then LMX1B is in the dorsal and ventral mesenchyme. And LMX1B itself is totally sufficient to establish dorsal pattern. So if you do this here and you now infect the limb bud with an LMX vector, wild type in cross section, the dorsal bones and tendons are very different from the ventral bones and tendon. But LMX, you get a double dorsal phenotype where the dorsal and ventral are completely symmetric because LMX is making both dorsal. So it's the dorsal phenotype. So the modern molecular understanding that you'll see in textbooks is shown here. It actually goes back one step further. Engrailed is in the ventral ectoderm repressing Wnt7a, which is the reason Wnt7a is only in the dorsal ectoderm, which establishes LMX1B here and so on. Each of these genes has been knocked out, and the results are completely consistent with the misexpression experiments. If you lose Wnt7a, you don't have LMX, you have a double ventral pattern. But the interesting thing is, if you knock out LMX1B, or it's called 1B now, but LMX1, if you knock out LMX1, which was done by Randy Johnson's lab, for an example, the only place you change dorsal ventral pattern is distally. The proximal limb, which also has Wnt7a and LMXB expressed in exactly the same way, does not change dorsal ventrally if you lose LMX1B. And this fact has been around since 1998, and it's totally ignored in the literature. And this is, people want to work on something. Nobody knows how the dorsal ventral axis is established proximally, why the dorsal is different from ventral. But when John thought he wasn't seeing changes proximally because he was transplanting the ectoderm too late, you couldn't do it early enough to flip to do the surgical experiments, it wasn't that the proximal was already specified. That interpretation was wrong. Something else is going on proximally, and we still have no idea what it is. So it's another thing that people can come back to. All right, returning back to John, another important line of investigation of Saunders at Marquette that also built on his graduate work involved cell death, as Carol mentioned to you earlier. The dye that was used to observe the AER was Nile blue. Nile blue is a vital dye. It's excluded from living cells and taken up by dying cells. And actually, the reason the AER shows up so strongly, there's actually a huge amount of apoptosis in the AER. It's rapidly growing and rapidly dying. So it's a sort of a turnover. And that's why the AER shows up with Nile blue. Well, John extended this and started looking at other regions that are dying using first Nile blue and then other methods. He identified multiple regions of cell death. This is before apoptosis was known as a distinct mode of dying. So he just called them necrotic zones. And he observed that there was some necrosis here in a place that had previously been described histologically as being opaque. And he realized that the opaque patch is actually between the radius and ulna and humerus, and it's a region of cell death. He also defined a region here at the proximal end, at the posterior side of the limb that he called the posterior necrotic zone, and cell death up here that he called the anterior necrotic zone. And so he defined a number of different regions of cell death. Now I wanted to digress just from a second from the science to point out that this person here, Lillian Saunders, was actually John's wife. And she was involved in a number of his critical experiments. Lillian and John were married for 69 years, both surviving into their 90s. They met when she was a graduate student at University of Oklahoma. And when he was an undergraduate, she was his teacher. She taught him histology. And they started going out and fell in love and got married. And so when he moved to Marquette, she found a job at Mount Mary College in Milwaukee. But while she was teaching biology during the school year, she and John both spent summers either at Mount Deer Island Biological Laboratory in Maine, or importantly for us, at the Marine Biological Laboratory here at Woods Hole, where she was assisting him. And like John, she had very good hands under the scope and did some very nice work. And also from an MBL perspective, she was later a board member of the Associates of MBL, and she was the person who played the critical role in raising the money and realizing the importance of establishing both the gift shop and the information center. So your gift shop and your information center at MBL, you can thank Lillian Saunders, who came with John to MBL and really appreciated the institution. Okay, returning to the science, they defined a number of places of cell death, including, as Carol mentioned, importantly, the interdigital cell death shown here. And the reason that it hadn't been observed before as cell death is because cell death is very rapid, but it's also cleared very rapidly. If you don't stain for it, you don't see it. If you just observe embryos, you can't see things rotting. It's seamlessly removed. And people did know cells died during embryogenesis before John, but a lot of the speculation was that it was just sort of turnover. John was the one who really pushed the speculation that cell death plays an important role in normal development of embryos. He was the one who hypothesized the function of cell death is to eliminate transient biological structures that are no longer needed for development, such as, for example, the interdigital region in the wing. Now, while this is obvious today, it was more of a novel concept at the time. The predominant view having been that some tissues had more cell death than others, it really was about tissue homeostasis. And the idea that the purpose of cell death is to eliminate transient biological structures that are no longer needed is really a sort of obvious thing today that's really attributable to John. Again, in the modern era, we know a lot about cell death in the limb, and we know about apoptosis, of course, at a molecular level, but we also know the things that trigger it in the limb. So for example, tunnel staining, which is a way of looking at cell death shown here in the interdigital regions, is triggered by a pathway that includes BMPs and MSX, and a large number of labs have contributed to that. And as was mentioned in the introduction, I'm very fond of evolution in addition to embryology, and changes in cell death pattern can play a major role in the evolution of structures. So I'll just give you a couple examples from the limb. One, the most famous one, is of the webbing of the duck. So here you see the apoptosis in between the digits in the chick that's going to lead to the digits being separated. In the duck that has webbing, there's a little bit of apoptosis at the tip giving you these notches, but for the most part, the webbing doesn't undergo apoptosis. Now, the genes I mentioned a couple seconds ago, BMP and MSX, are expressed interdigitally just fine. So you have interdigital cell, you have the genes that drive interdigital cell death, but that doesn't happen in the duck. And the reason it doesn't happen in the duck, as Juan Hurley first showed, is that there are BMP antagonists such as gremlin that are expressed on top of the BMP in the duck that are not expressed there in the chick. So the BMP pathway is blocked, and because of that, you don't get the same level of cell death. So that's an example of prohibiting cell death. There are other cases in the limb where you expand cell death. An example of that comes from the horse. So this is a cartoon of horse evolution. Of course, the ancestral horse had multiple digits. It had 4 digits, the earliest Eohippus or Hyracotherium, the dawn horse. It then went to 3 digits and ultimately just to a single digit, while went from 4 to 3 metacarpals or metatarsals, which fused to form the cannon bone. So how do you get from multiple digits to a single digit? Well, initially it was not looked at in the horse, but it was actually looked at in a rodent, because of course all of these intermediates are extinct and the horse is not a great lab animal. But the rodents are good lab animals, and the Jerboas, this group of animals here, have recapitulated a lot of what happened in the horse. So you can see here, there are 4-toed members of this family, 3-toed members of the family. They never get down to a single toe, but they go from the 5 to 4 to 3 toes. And they also form the cannon bone go from 3 metatarsals to a single cannon bone. So how do you get from multiple toes to a single toe, or from multiple toes to 3 toes? Well, in the Jerboas, it was shown the way that happens by this expansion of cell death. So this is one of these genes that drives cell death, MSX2. In the Jerboa forelimb, which is very similar to the mouse forelimb, you don't see an expansion. But in the mouse hind limb, you don't have much cell death. In the Jerboa hind limb, where the fifth digit would have formed and where the first digit would have formed, you have massive expression of MSX2, which leads to massive expansion of where the cell death occurs. So where the limb buds would normally start forming the first and the fifth digit, the digits are basically just chewed away by cell death by an expansion of this program of interdigital cell death. So that you pattern all 5 digits, but you carve out those 2. And now that you know what's going on, you can return to the horse. The horse is, of course, a beautiful animal, and one wouldn't want to be doing things that are in the mouse or the jerboa to collect embryos. You have to kill the mother. You wouldn't want to do that with a horse. But there's a trick of sort of like an unexpected twist to the embryology of the horse that makes it very easy to examine these early embryos. And that is, in the horse, it doesn't, the blastocyst comes down through the uterus, through the fallopian tubes into the uterus. But when it hits the uterus, it doesn't implant right away, whereas the human or mouse embryo immediately implants into the uterine lining. The horse embryo first undergoes fixation, where it attaches to the wall of the uterus, makes sort of a connection for blood transfer after about 16 days of fertilization. But it doesn't start implanting until 40 or 45 days, and it doesn't get totally embedded into the uterine wall until 140 days of pregnancy. So when they want to do embryo transfers, for example, for racehorses and so on, all they do to harvest the embryos is shoot some saline down the birth canal, and the embryos come right out and the mother just keeps eating hay through the whole process. It doesn't hurt her at all. So it's very easy to get these sorts of horse embryos if you collaborate with a vet school. And having those embryos now looking at the right stages, you see the exact same thing that you saw in the Jerboa. This is MSX2. There's a single digit. The other digits were going to form initially, but there's massive expansion of what would be the program of interdigital cell death, leading to cell death above what would be the other digits, so you only leave the central digit in the absence of cell death. Well, John, when he was looking at cell death, was interested in things such as when is the cell death specified? So he would take regions that were fated to undergo cell death at various stages and graft them elsewhere in the embryo and say, if you do this graft early or late, will they die in the new location? And he found that from Hamburger-Hamilton stages 17 to 21, looking at the posterior necrotic zone, that is this one down here, if you graft between 17 and 21, they don't die. But if you graft between 22 and 23, they have the same type of degeneration that normally starts at stage 24. So he could define the time when the cell death was to be specified. The other thing he would decide to look at, we said earlier that if you remove an AER, you have massive cell death underneath the tip of the limb bud. So the AER protects cell death. These cells are dying. Which is dominant? If I take cells that are fated to die and put them under the AER, will the AER rescue them? So that's this experiment here. You take cells that are fated to die and graft them at the tip just underneath the AER, what happens to them? Well, the answer is they don't die, but he got a much more interesting result than that they just don't die. The reason he did the experiment was to look at cell death, but what he got was kind of mind-blowing because this tiny group of cells there's a very small group of cells, and they could be, and even you could actually irradiate them before transplanting them. They don't expand into anything. But if you transplant them, they're capable of causing this enormous expansion of the limb bud, giving you polydactyly, multiple digits. But more importantly than polydactyly to John is the orientation of the digits is reversed. Whereas the anterior digits, like digit 2 is shown here, are in the middle, the posterior digits are now on both sides. That is, it's as if the closer you are to this region of cells, the more posterior you are. If you put the cells over here, the digits that form at this extreme of the polydactyly are posterior. And of course, the endogenous cells here make these cells posterior. So the ability to polarize the limb, which he thought was the most significant part of this, gave rise to the term the zone of polarizing activity. So this is, now John has defined the key activity for defining the proximal distal axis, that is the AER, the dorsal ventral axis, that is the dorsal ectoderm, and now the anterior posterior axis, that is the ZPA. He nailed all three of them, and his interpretations were spot on. So as soon as this was found, lots of people became very interested in how this might work. For example, Cheryl Tickle, who worked with Louis Wolpert, did an important experiment where she grafted different numbers of ZPA cells. And the more ZPA cells you graft, the more dramatic the duplication. Just a small number of cells would repolarize the limb. That is, you get the mirror image duplication. But if you have just 100 cells, the only duplication is of digit 2. With 500 cells, you duplicate 3 and 2. And with 1,000 cells, which is really the full ZPA at this stage, you duplicate the whole limb. And one way of interpreting this would be in terms of a morphogen. So a morphogen is a secreted protein that is interpreted at threshold levels. You can imagine the ZPA makes a factor that is secreted and diffuses across the limb bud. It gets more dilute the further away it gets from the source. And at various thresholds, you have instructions. At high levels, you make digit 4, intermediate levels digit 3, and low levels digit 2. And of course, if you graft another potent source, you get high levels at both edges, then you get lower levels and then still lower levels, and you get the full duplication. Whereas if, as Cheryl did, you only grafted a few cells, you only hit the lower threshold. You never make enough to hit the higher threshold, and then you get this mirror image duplication, but only of digit 2. So that's at least one interpretation that you can have of it. Another important finding was that once people found the ZPA caused these duplications, they said, what else can cause duplications? So they tried grafting other things into the limb bud. No other region of the limb bud would cause these duplications, but there were a few other parts of the embryo that could. For example, grafting, as was shown by Gregor Eichley's lab, Wagner et al., if you graft a bit of the floor plate or the notochord of the neural tube, either one, a graft of the floor plate will give you mirror image duplications very much like the ZPA, which you would interpret as meaning that the floor plate makes a similar or the identical signal to the ZPA. So now jumping ahead to the modern days, the gene that we think is the most significant in this pathway is called sonic hedgehog. First cloned in the chicken by Bob Riddle in collaboration with Andy McMahon and Phil Ingham. And not only is it expressed in the posterior limb, it's also expressed in the notochord and the floor plate of the neural tube. This is actually the first in situ that was ever done with sonic hedgehog in a chick. So it's sort of, Bob does the in situ, and the importance of what the signal might be just jumped out at him. Not only is it in approximately the right region for the ZPA in an early limb bud, but the ZPA had been carefully mapped by Somerville's lab in 1985, where at different stages of limb development, you could see which tissues within the limb bud, if you graft all the different, sort of cube up the limb bud, dice it up, and graft each dice piece into the anterior and say which piece can give you a ZPA-like effect. Both the extent, how effective it is, this is 19% effective versus 100% effective, and the location at every stage really perfectly matches where sonic is expressed. Moreover, sonic is completely sufficient to cause such duplications. If you take a bead soaked in sonic hedgehog protein, put it in the anterior of a limb bud, you get perfect ZPA duplications. And as Cheryl showed, if you put in less sonic hedgehog, you get less of a duplication. So that seems to be working in the graded response. And not only does it seem to be working in a graded response, as Andy McMahon showed, if you take a look at sonic hedgehog protein is in a diffuse gradient, even though the messenger RNA is very localized. So all that's consistent with this morphogen model. But it's not the only model that's out there. There are other ways that you can view this. For example, Cheryl Tickle had very strong data saying that there might be a temporal aspect to it. If she marked cells right next to the ZPA and then left the ZPA and then removed the ZPA, or in the anterior put in a sonic hedgehog bead and then remove the sonic bead, after a short period of time, with just a little bit of the exposure, she would get a duplication only of the first digit. If she left the hedgehog in for a longer time, those same exact cells would get turned from a first digit into a second digit. And those same cells, if they saw sonic long enough, would develop into the most posterior digit. So in this case, the axis isn't sonic concentration, but it's sonic exposure in terms of time. And she argued that it was a temporal length of exposure. That's coming from chick experiments. In the mouse, Sue Mackham first showed that by conditionally removing sonic at different times in the mouse, that if sonic acts, at least in the mouse, if it acts as a graded morphogen, it has to do so very early, because early, all the patterning seems to happen in the mouse over a very short period of time. Sonic is needed subsequently, but it's needed for the expansion of the tissue and the growth of the digits. It's not required for their specification. Most recently, Susan now has data that says, well, if this is the right time frame, the interesting thing is that that time frame, if you look at which cells are responding to sonic, only a few posterior cells are responding. By the time the anterior cells even see sonic, it's too late for it. It can cause their expansion, but it's too late for specification. So if sonic isn't reaching all the cells, then something else must be reaching the cells. So she's proposing a relay mechanism. And this is really a very open question. We really don't know what's going on at all in terms of, I shouldn't say we don't know at all, but it's very much debated exactly how hedgehog works in defining the limb. So the last thing I was just going to tell you, and I know I'm running out of time, so I'll go through this quickly, is that sonic is conserved in its function, not just in chicks, but in mice and in other mammals. And you can have mutants, for example, preaxial polydactyly, that look like a mirror image duplication of the thumb, the first digit in humans, very similar sorts of things to what you have with a low concentration of sonic hedgehog. And those have been mapped, and the mutations in both humans and mice that cause this map to a regulatory region that's a long distance regulator of sonic hedgehog, the ZPA regulatory sequence, or ZRS, which is in an intron of an adjacent gene. And if you take a look at the mouse, why this mutation causes these slight duplications is because in the mouse, you get a little leakage of just a little bit of sonic in the anterior, which causes just this little bit of duplication. And there are also total knockouts in the mouse. Of course, if you knock out the whole gene, it's very lethal because it's very pleiotropic. But if you knock out just the enhancer that drives sonic expression in the limb, they're naturally occurring in the mouse. You just only affect the limb and you get a single digit. And that's exactly what you get in the chicken and a naturally occurring mutant that has a mutation specifically in the ZRS enhancer called oligozugodactyly. And then there's a human mutation that also causes the same thing. So John's insights were not only important for understanding limb development, but also important for limb pathology in humans. I've sort of quickly gone through giving you a taste of John Saunders' discoveries and some of the more recent insights that his earlier work made possible. But not only his scientific discoveries, but his rigorous interpretations that were made long before genes were identified have really stood the test of time. He was a truly astute experimentalist, a gifted writer. His papers are a pleasure to read. He was a committed mentor and educator, and an absolute scientific giant on whom a lot of the current work still continues to build. So I'm really pleased to be able to introduce him to you today as the father of limb development and a very worthy person to have as the Endowed Lecture named for. So I'll stop there. Thank you very much.
Thank you so much, Cliff. That was really fantastic. We don't seem to have any questions in the chat, so I'm going to let students from the course come up and ask questions.
Hello, my name is Guilherme. I'm a PhD student in Wisconsin-Madison here at the course. It's very nice to see this historic perspective and the classic studies, and just got me thinking, what do you think are the most promising future directions in the modern studies of limb development? What areas we still don't understand very well about how the limb is patterned?
So I think I highlighted a couple places along the way where we still don't understand patterning. I think there's a tendency, the textbooks show sort of simple diagrams that make some sense about patterning, and people sort of say, oh, okay, we understand patterning. We really don't understand it in a very serious way. I think that right now, if I were to show you the anatomy of a human limb, you would see there were dorsal muscles and ventral muscles. And for example, your bicep has 2 heads, your tricep has 3 heads. Why does the bicep have 2 heads and the tricep have 3 heads? Nobody knows. No one has a clue. And yet if I can't explain that, if I can't tell you why the bicep fibers are going along the length of the limb and not horizontally, which we don't know, or why there's even a bicep forming where the bicep forms in the first place, which we don't know, then I would say we don't really understand limb development very well. So, and similarly, the skeletal elements, I mean, it's a lot of the same thing. There's some idea of how there's proposals for reaction diffusion mechanisms for forming the different digits, which you may or may not know about, but we don't have time to go into. But there seems to be some Turing type mechanism giving you digit, non-digit, digit, non-digit, a pattern upon which the ZPA's effect is overlaid. But that gives you the digits. What about the more proximal limb? How do you get from why 1 bone then 2 bones? Why do you at the elbow why at the elbow? What's the discontinuity there? But then what gives you 1 bone then 2 bones? There are Hox genes that are expressed in the different limb segments. But if you knock out the Hox11 genes, which are expressed in the lower arm, you don't get a single bone. You get tiny little nubbins of 2 bones that never grow. Yeah, the Hox genes are critical for making the zugapod, but how you get from 1 bone to 2 bones is totally unknown. So I would say there's a lot of things we don't know in terms of how the elements form. We also, the roles, my lab's done, I'm not going to talk about it all today, but we've done some work in the gut on physical forces. There's very little work on physical forces in the limbs. All of embryology is sort of an interplay between signaling and the morphogenic properties of the mechanical forces that surround the tissues. So how do you actually get the shape of the limb? How do you get, your palm is flat. How does the palm get so flat? What's going on there? It certainly involves physical forces. So I would say that, the limb is described as sort of a classic system that's been classically described and classically understood, but that just gives us a platform for asking questions. I think there's a huge amount we don't know.
Cool. Thank you very much.
Other questions?
Well, while someone else is coming up, the other thing that we also don't know, there's still a lot to be done on the evolution of the limb, something that our lab's also interested in.
I got to tell you, I'm going to go get horse embryos and study neural crest and forest stuff. I'm really excited by this. We have another student here to ask a question.
Hi, my name is Kara. I'm a grad student with Andrew Murray and Sean Eddy, actually. I just have a clarification question. In both the ZPA and the AER, I thought you were saying that the cells that are signaling are also the ones that are either currently dying or are fated to die. Is that correct? And if so, can you explain what the order of events there is?
So the ZPA and the cells that are fated to die overlap. I'm not going to go back to the slides. I showed a map at one point where I showed that Sonic Hedgehog matched the location of the ZPA at every stage of limb development. That the region of the ZPA shifts over time. It starts out being very proximal, but it always is sort of just below the range of the AER at the posterior edge. The cells that die are very proximal cells. The posterior necrotic zone is a very early group of cells that overlaps with the ZPA, but they're dying after the ZPA sort of moved on, if you want.
I see.
And the more distal part of when the ZPA is there at a stage 26 or 25 limb bud, those cells never undergo apoptosis.
Okay.
And if you fate map the ZPA using a Cree-lock system with a sonic specific promoter and a flox marker like GFP or something, you find that the ZPA contributes to both in a mouse to the 2 most posterior digits, both the little finger and the ring finger. So those cells stick around. In contrast, the AER, those cells are constantly turning over. And if you fate map them, they actually are all gone, because eventually when the AER ceases to exist, late in limb development, they completely undergo apoptosis. So there is apoptosis connected with both, and certainly apoptosis, the connection of apoptosis is the reason John found the different tissues, but it was more, I presented it in the talk more as a sort of intellectual link, just tracing how he made his discovery rather than really being something about cell death and morphogen signals per se.
So you don't think, I was just going to ask, like in terms of especially the turnover rate, is the rate of turnover thought to be related at all to the fact that they're signaling, or do you think it's just kind of independent?
It's something that's totally ignored. I've never seen anything on the regulation of cell death within the AER, or why it occurs, or how it's regulated. It's just not something that's looked at. And I mean, I also glossed over in my talk, the AER actually has a dorsal half and a ventral half to it. So it's the seam between the dorsal and ventral extremes, but there's at least one gene that's expressed only in the ventral half, engrailed actually, in the ventral half, but not the dorsal half. And what's happening at that boundary, how that boundary critical for AER formation. There's a lot that we don't understand about the AER, including cell death. But it's sort of, again, back to the fact that we have a superficial understanding. I think I wouldn't say that's the limbs. It's not that the limbs sort of become a backwater where we don't understand things deeply. I would say for morphogenesis in general, we don't understand things deeply. And part of that may be that we're pushing the edge of single gene interrogation. Maybe that we're getting to the point where to get further with things, in some cases, you have to go to a systems approach.
Thank you.
Okay, we have one last question for the webinar.
Hey, very nice talk, Cliff, and good to see you. I give lectures on limb patterning, and I always start off by showing it isn't amazing how our arms are the same length, and then I have nothing after that to share. So what are your thoughts? Like, what ensures the robustness of things like arm length?
Yeah, so that's something that Louis Wolpert was famous for doing, that he would address students and say, stand up, stick out your arm, put them together, close your eyes. How come with your eyes closed you have them at the same? So that was one of Louis Wolpert's favorite examples. So we had some thoughts about feedback loops that might be I mean, there's got to be some self-regulation. If you haven't grown enough, you grow a little bit more, because I mean, you're absolutely right. The limbs are very much the same length. And not only are they the same length, but you can do a fair amount of hacking early on and they still end up being the same length. Now, if you do enough hacking, you can end up with a dwarfed limb. But there's got to be some feedback loops that are, and what you're doing is you're running the same sets of feedback loops that are governing how, if there's too much proliferation, the proliferation signals go down. If there's too much proliferation, too little proliferation, the proliferation signals go up through some sort of buffering system that must be operating independently in the two limbs, but with parameters that are specific for the individual based on the gene expression, the exact enhancers and gene context of the individual. So for any given individual, they work equivalently on the two sides, but it has to be very robust. I mean, it doesn't really matter so much that our arms are the same length, but if your legs aren't the same length, and if you're a tetrapod, if your two forelimbs or two hind limbs aren't the same length, there's an enormous, the selective disadvantage of being hobbling in the wild with predators is just enormous. So, and because of that, I'm almost sure that it's not a simple answer, because it was so important and critical to get them to be, the left and right to be exactly the same for biomechanics. There must have been many layers of selection and many layers so that multiple genes. And because then the corollary would be, if there are lots of genes involved and lots of processes involved, it's unlikely to be something you get at with a very simple, oh, look, there it is.
Yeah, it'd be hard to find a mutant in it. Yeah, exactly.
Exactly.
Okay, hey, thank you so much.
Yeah, because you want robustness in spite of occasional mutants.
Okay, Cliff, thank you so much for an absolutely fantastic Saunders lecture. We've been thrilled to listen to what you've told me today. Let's give Cliff another round of applause. I believe that's the end of our broadcast webinar. And so we will log off, and then Cliff, we're going to take a 10-minute break here at Embryology and join you again for your second lecture.
Are you going to send me a different link?
I believe John Young has already sent it to you.
Okay, I'll get on email and look for it. Thanks, John.
Thank you very much.
Bye-bye.
All right. Thank you, everyone, for attending. We hope that you will consider registering for other lectures in the series. Also, our Friday evening lecture series has gone virtual once again this year, and we hope that you will consider registering for Dr. Ann Harrington's lecture that will be taking place on June 18th. Thanks again, and we look forward to seeing you all in the future.